So in today's lesson, we will start to explore the process of gene expression. Okay, whatever information that is coded in the gene, in the cell, how is the information being expressed into the form of polypeptide in the cell? So therefore, it involves a few steps. You're going to look at it step by step. So first thing we want to know the gene codes. The gene that codes, it is the gene that codes for polypeptide, and how the genetic code for polypeptide. So how does genetic code look like, and how does it code for the polypeptide? And then we will also look at transcription. Okay. 1, 2, 4 is the learning outcome. That's why I skipped number 3. Okay. Because number 3 is protein synthesis, which we will not look at today. So therefore, it's learning outcome 1, 2, and 4 first for 6.2. Okay. So let us look at this term gene expression. Okay. So when you want to express yourself, there's something that arises in your mind, right? A thinking, a thought arises in your mind, and then you say it out or you act it out. So this is called expressing yourself, right? Whatever thought that you have, you express it out. So gene expression means whatever code that the gene has is being expressed out, means it's being converted to a polypeptide, which the gene codes for. So let us look at gene expression. So gene expression means that the gene, which is found in DNA, is converted to polypeptides. Means the code in the gene is converted to polypeptide, not protein. Protein is secondary school level, okay? SPM level. So when you go to A levels and higher level, the gene actually code for a polypeptide, not a protein. Because you know that a protein can be made up of one polypeptide or more than one. So if it's more than one, that means it's more than one gene. So more accurately, a gene codes for a polypeptide. Okay. So this gene expression is based on the central dogma. Dogma means principle. Principle means the way the things work. Okay, the way the things work is called principle, right? Okay, so central dogma, central principle. So what does the central dogma say? Central dogma says that DNA codes for the synthesis of new DNA by replication, as well as RNA by transcription. So DNA in the cell code for the synthesis of new DNA, as well as the synthesis of RNA. Synthesis stands for to produce. Okay, the word synthesis stands for to produce. Okay. And then the central dogma also says that mRNA codes for the synthesis of polypeptide. So you use mRNA for the synthesis of polypeptide by the process called translation. Okay, so translation is polypeptide synthesis. Transcription is RNA synthesis. So this whole process is called the central dogma, right? And the direction how things work. So polypeptide does not code for the synthesis of mRNA or polypeptide or DNA. So you can see the arrow shows the central dogma is always in one direction. DNA is used to synthesize new DNA. DNA is used to synthesize mRNA. mRNA is used to synthesize polypeptide. So it's never the opposite direction. So that is the central dogma. Okay. So
teacher.
Yes.
Can you let Chu Jiaying in? She's waiting for you to accept her actually.
Okay, Chu Jiaying. Yeah, I think someone is outside already. Yeah. Thanks for informing me. Chu Jiaying. Okay, thank you. Right. Okay, so gene expression involves 5 stages. So this would be your homework. you need to fill up all the names and the name of the molecule, the name of the process in this diagram in your notes. Okay, I've modified the notes yesterday because I think it's very important for you to write it yourself. Then you remember better. Okay, so we start off with so the process that will occur in the nucleus first. So we have got DNA, a lot of DNA in the nucleus. So every DNA will have got genes in it. So gene is a specific length of DNA. So see, this whole thing is a DNA. So in this particular length of DNA, you have a gene, right? And then in this particular length of DNA, you got another different type of gene. So gene is considered as a length of DNA. All right, so I'm magnifying this part of the gene to show that this gene is actually made up of DNA, which is double-stranded, and there are complementary base pairing between the complementary strands. So this is a gene from the DNA. Okay, so from the gene, transcription will happen. Now, the word transcription comes from the word transcript. Now, transcript means for a copy of. So when you take your book or document and you go and photostat it, right, or you scan it. So the outcome of the scanning is the copy that you have is called the transcript. Why? Because they look similar. Okay, so transcription is a process where the codes on the gene is converted to the codes on the RNA, right? So that's why it's called transcription. So the first RNA that's formed is called the primary RNA transcript. Primary means the first one, okay? Transcript means a copy of. So why is it a copy of? Because the gene is made up of base sequence, right? Or you can call it as a nucleotide sequence. Okay, right? Base sequence or nucleotide sequence. So when you talk about the base sequence, it means you're referring to the base over here, right? Whether it's A, T, or G, or C, so they have got their own base sequence, right? Now, if you say nucleotide sequence, is also correct because you're considering the whole monomer, including the base, right? The whole monomer, including the base. So you can say nucleotide sequence or you can say base sequence, right? So you're converting the base sequence of the gene, okay, to the base sequence of the RNA. Base sequence or nucleotide sequence in the RNA. So it's the same language, it's a base sequence kind of a language. So therefore, this is called a copy. Because from base sequence, you copy it to base sequence. So it's a copy. So it's a transcript. Okay? So after that, can you see that this strand here, the primary RNA transcript versus this strand here, can you see this strand here is shorter? So that means from here to here, a small segments have been cut away. So this is called processing. The second step is we need to process the primary RNA transcript to make it shorter in order to make the mRNA, or can make it tRNA or rRNA. Okay? So depending on which part of the gene we're talking about. So after that, the completed mRNA, tRNA, or rRNA will be transported to the cytoplasm. So now the rest of the process will occur in cytoplasm. So tRNA, mRNA, and rRNA in the ribosome, okay, will be transported through the nuclear pore to the cytoplasm. So next step is translation. So translation is where your mRNA will be converted to polypeptide chain. So this process, translation, you already know translation means you change the language, right? You want to translate the language, let's say from Korean language, okay? From Korean, you want to translate to English. Okay, from Japanese, you want to translate to English. So this is called translation. So why is it called translation here is because the base sequence, right? The base sequence or the nucleotide sequence is converted to amino acid sequence. So it's a different language already. From a language of base sequence, it becomes language of amino acid sequence. So that's why this process is called translation. Okay. So with that, you will not mix up the difference between transcription and translation because of the meaning. Right. Of course, another way to remember it is transcription is a C. C comes first, followed by S. Okay, C will come first, followed by S. So this is another way for you to remember. Okay. So after that, you see the polypeptide chain. How does it modify itself to become a protein? Is polypeptide chain will then step number 5, modification to protein. So it can fold into secondary or tertiary structure. Now, this is my short form for secondary tertiary structure. So it can fold into secondary tertiary structure, or it can join with other polypeptides to form the quaternary structure. So it can join with one or a few polypeptides to form the quaternary structure. It can also form complex with non-protein components. That means it combines with non-protein components like iron, Fe2+, and porphyrin ring to form the hemoglobin molecule. So that will be our completed protein molecule. So this entire process here from number 1 to number 5 is considered as the gene expression. So whatever that the gene code for is being expressed to the form of the protein eventually. Okay, so this is gene expression. So let us look at what is a gene recap. So gene is a length of DNA, okay, a length of DNA that codes for a specific polypeptide chain. So for example, if this gene is supposed to code for insulin, so this gene will only code for insulin and nothing else. Okay, if this gene is supposed to code for glucose carrier protein, so it will only code for glucose carrier protein. So it's very specific. Okay. So the length consists of a specific sequence of nucleotide or specific sequence of bases. So every gene got their own specific base sequence or nucleotide sequence. So, and this gene will determine the structural, physiological, and biochemical characteristic of an organism. So which means to say that the gene code for these properties of an organism: structural, physiological, and biochemical. So structural means whatever physical form that you can see, right? For us, like 2 eyes, 2 ears, nose, okay, smooth skin, okay, hair, and 4 limbs, and so on, right? So this is called structural, which you can see. Physiological means how our body functions. So how do we obtain oxygen from the air? We use lungs. Whereas for the fish, they use gills. Whereas for the frogs, they use their moist skin, right? So whereas for plants, maybe they have got stomata. So the air from outside can go into the leaf, right? So this is called physiology. How do we move? We have got bones and joints and muscles, so that help us to move around. And then we've got nerves as well to transmit the action potential. So how our body function, how our body remove excess water, we have got our kidney. How our body detox, we got our kidney and liver. So this is called physiology. Biochemical means all the chemical processes in our body, like respiration, like deamination, like formation of urea, okay, a conversion of glucose to glycogen. So these are all biochemical. So basically, all these properties of us, they are all coded by the gene. So can you see, it's so wonderful. Genes are just DNA, made up of bases, and all these bases can eventually code for all these properties that we have in our body. So it's actually, our body is actually very, very wonderful. As you can see, it's a miracle. So it is a miracle, right? If you are being born, it's a miracle, right? So you need to treasure your life, right? And be grateful for that. So each living cell carries the full set of genes, right? All the cells in our body, almost all of them, except for our gametes, carry the full sets of genes, right? But however, if all the cells carry the full sets of genes, it means all our cells are genetically identical. Why is it in our body we can have got different cells even though the genes are the same, right? It's because in different cells, different genes are being switched on at different times. So even though we carry all the genes in all the cells, but not all the cells will express all the genes. They will only express the genes that they need, all right? So therefore, liver cell will only express the gene where they will remove the alcohol that we drink. Say, for example, okay, liver cell, correct? Liver cell? Yes, liver cell. So liver cell also will express the gene which will code for enzymes to convert glucose to glycogen or to break down the glycogen to glucose, right? So because that's the function of what the liver cell is supposed to do, all right? So that's the example. Different cells, we have got switch on different genes at different time when it needs. So your alpha cells and beta cells in your pancreas. So your alpha cells will switch on the gene to produce insulin when you need insulin. Sorry, the beta cells, okay, the beta cells. So alpha cells will switch on the gene to produce the glucagon only when you need glucagon, right? So therefore, different cells in the body will switch on different genes at different time based on what cell it is, so what function is supposed to do, what kind of proteins they need to do the function, okay? So the specific sequence of bases in a gene will be converted to specific sequence of bases on mRNA. So this process is called transcription, means making a copy of. So how does this process occur? Is by complementary base pairing. So how do you copy the gene into the mRNA? Is by complementary base pairing. Okay, so let us look at this, this diagram. So now we have got one of the two strands of DNA. So only one of the strand will act as a template. So that is called the template strand, okay? Or the transcribed strand, the strand that will be transcribed of the DNA double helix. So the complementary strand is called the non-transcribed strand of DNA. Okay, so I've shown you the base sequences here. So in your new notes, you need to draw the mRNA that will be synthesized from this template strand of DNA. Okay, so that will be your homework as well. So now, after transcription, what we'll expect to see in the mRNA, Rebecca, tell me what is the base sequence you expect to get for the mRNA? Rebecca.
AGT.
AG. So AGT is the base sequence that is found on the DNA template strand. So if I want to transcribe it to my mRNA by complementary base pairing, what should be the bases that's supposed to find here in my mRNA?
E.
Again?
ECA.
E stands for E, is it?
TT.
T.
TA.
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 TATA again.
No, TCA.
TCA.
Yeah.
Some more?
A.
A.
T.
T.
G.
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 C.
T.
C. C.
C.
Yeah.
Okay.
T.
T.
A.
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 G.
G. So is this correct, Rebecca?
Yeah.
Okay. Actually, I want to ask someone else. Can someone else tell me that is this correct? You can type in the chat box. Correct or no? If no, then you must tell me what is correct. Tan Pei Wen. Okay. Tan Pei Wen says that it's supposed to be UCA. Tan Pei Wen. It's UCA, correct? Okay, it's UCA. Now, you see, Tan Pei Wen says that this is supposed to be UCA. Okay, UCA. So that means the first one is U and not T. So which one is correct? TCA correct or UCA correct? Class, please type your answer. UCA. Yeah, UCA. Okay, yeah. So you see, this is where a lot of students may actually make mistake here, is when we convert DNA, all right, to RNA. So we know that in RNA, instead of a T, it is a U. So normally A pair with T, right? But now because this is RNA, instead of T, it is a U because RNA does not use the T. There's no T in RNA at all. So therefore in RNA, instead of a T, it is a U. So this is something that all of you must learn, okay? Because it's very common that students are asked to do the complementary base pairing and then to decode whatever that's given there, all right? Okay, hang on. Okay, so therefore it's supposed to be UCAA, and then this is also a U here, okay? And the T here is also a U. So that will be our mRNA. So this is a homework that you need to draw into your notes. So draw the basis of the mRNA that will be synthesized from this DNA template strand, okay? So therefore, thank you everyone for participating, all right? So therefore, this will be the base sequence of the mRNA. Now, after that, this mRNA is used as a template to synthesize the polypeptide. So this is called translation. So the sequence of bases is translated to the sequence of amino acids on the polypeptide. So how do we translate it is now I'll show you first. Later on you learn, okay? So in the original DNA template strand, every 3 bases will code for 1 amino acid. So these 3 bases here, AGT, code for 1 amino acid. So these 3 bases is called a triplet code because the code is carried in the DNA. Triplet because it's 3 bases. Okay. So then in my mRNA, these 3 bases will code for the amino acid. Now these 3 bases is actually transcribed from these 3 bases. So these 3 bases is called a triplet codon. So these 3 bases in the mRNA is called the triplet codon because the code is in the DNA. So once you copy it from DNA to mRNA, we no longer call this a code, we call this a codon. Okay, a codon, because the code originally is found in only the gene, which is the DNA. So when it's in mRNA, we don't call it a code anymore, we call it a codon. Okay, triplet codon. So from triplet codon, we can use a table to find out what this code for. So this code for amino acid. Okay, so this UCA code for serine. So remember the short form of an amino acid can be represented with a 3-letter word here. So serine. So AUG code for methionine. GCU codes for alanine. AAG code for lysine. So this is what you need to write to your notes, which is empty, yeah? Okay, and all this also you need to write, okay? So how to convert the genetic code that is found on the DNA template strand to amino acid sequence in the polypeptide chain? So this is wonderful, right? So you want to go and actually break the code, the secret code in our DNA, right? So these are the features or the properties of the genetic code. So features stands for properties, okay? So what are the properties of the code? Number one, the code is a triplet basis. Number two, it is universal. Number three, it has got this degenerate property. Number four, punctuated. Number five, non-overlapping. So the codes do not overlap. So what does it mean? So number one, now this diagram on the right-hand side is an mRNA genetic dictionary. So it's called genetic dictionary. So dictionary means you want to find out the meaning of the word, right? So over here, genetic dictionary means you want to find out the meaning of the codon, okay? Because this is mRNA. Can you see? There's no T inside here. All of them are U. So that means these are the triplet codon found in the mRNA. So that's why it's mRNA genetic dictionary. There is also a DNA genetic dictionary, which shows the genetic code. Okay, it shows the genetic code. This one shows a genetic triplet codon. Okay, so what does a dictionary do is that it tells us the meaning of the word. So here it tells us the meaning of the triplet codon. That means this triplet codon, for this triplet codon, what does it code for? So what is the amino acid that it codes for? You can see here, UUU or UUC code for phenylalanine. Right, UUA, UUG code for leucine, right? CCU, CCC, CCA, CCG code for proline. CAU, CAC code for histidine. So this is the genetic code, all right? So number one, the genetic code is a triplet code in DNA or a triplet codon in mRNA. So that means triplet means 3 bases. So 3 bases on the DNA or RNA codes for 1 amino acid in the polypeptide chain. Okay, so a DNA dictionary will show the amino acid coded by the triplet code, and an mRNA genetic dictionary will show the amino acid coded by the triplet codon. All right, so how do you read it? Okay, now this is a first position here, second position, and third position. So if I've got a UAC, what does UAC code for? Charmaine? Charmaine, what does a UAC code for?
Is it trypsin?
UAC code for trypsin. Trypsin. Yes. This is actually not trypsin, this is tyrosine. Trypsin is TRP, but it's correct. Okay. Thyroxine. Okay, correct. It is thyroxine. 2 marks. So this is how you read the genetic dictionary, okay? Right, next one. So now the next question is, why is it 3 bases code for 1 amino acid? Why I cannot use 1 base or 2 bases or 4 bases to code for 1 amino acid? Why must it be 3? So this is where you can see that the nature is so wonderful. The nature, that how it designed the living thing is so wonderful. Okay, you see, So why not 1 or 2 bases code for 1 amino acid? Why must it be 3? Now the tips here is how many different code can be formed using 1 or 2 or 3 bases to code for 1 amino acid. Okay, let us look at this one. Now in our body, we have got at least 20 different types of amino acids, right? So therefore, we must be able to make 20 different types of a code from our base sequence in the DNA in order to code for the 20 different type of amino acids that we have, right? So now let us look at this. Now, if we use 1 code to code for 1 amino acid, we only got 4 different types of bases. So therefore, if we use a one code to code for one amino acid, can you see here? We can only have got A, G, C, T. So therefore, we can only make 4 different types of codes if we only use one code for an amino acid, right? So which is actually not enough to code for 20 different types of amino acids. What if we use 2 bases to make the code? So that means the code is made of 2 bases. If you use 2 bases, you can see from here, altogether, there are only 14, right? 14 different types of codes possible if we use 2 bases to make the genetic code. So again, 14 is not enough to code for 20 different types of amino acid. So therefore, if we use 3 bases to make the genetic code, you can see from here, if we use 3 bases to make the genetic code, altogether, here there are 64 different types of possible codes based on the based on the different combination of bases in the triplet code. Then this is enough. This is more than enough to make 20 different types of amino acids. Can you see that? Okay, so because of that, our genetic code is always triplet code, because triplet code, then there's a 64 possible different types of codes, and therefore it's more than enough to code for our 20 amino acids. Okay, now this is a very popular question in MCQ, right? So this is a table where you need to fill in the blanks in your notes. Okay, you need to fill in the blanks in your notes for this table. So here we are comparing if you use 1 base as the code versus 2 bases as 1 code versus 3 bases as 1 code. And there are 4 different type of bases we can use to make the code. Okay, we've got 4 different type of bases. So what are the possible number of genetic code that we can make out of this? Okay, if we use 1 base for the code or 2 bases for the code or 3 bases for the code, where we have got 4 different type of codes, bases that we start off with, right? So this is a popular question in exam. How do you find out? You must use a mathematical formula. All right. So if you use 1 base, then how many number of different type of codes is possible? It's 4 power 1, which is actually 4. So not enough for 20 amino acids. If you use 2 bases, the number of possible genetic code is 4 power 2, which is 16. If you use 3 bases, it's 4 power 3, 64. Different type of codes. So it's enough for 20 amino acids. Now, this is a formula that you need to know and be able to use. So there's an easy way to remember this formula. Okay, it's actually a probability formula. Now, why? Now, imagine you got a bag. So you have a bag, and inside your bag, you start off with 4 different type of bases, which is A, T, G, C. So you need to use these 4 different type of bases to make your genetic code. All right? So now this is if your genetic code is made of 1 base. That means at any one time, you put your hand into this bag and you randomly pick 1 base out of this bag. So your base can be A or T or G or C, right? So therefore, if you only need 1 base, that means the formula that you're supposed to use is your bag has got 4 different bases and you only want to pick out 1 base at a time to form your genetic code. So it's 4 power 1. So there are 4 possible different type of codes you can get out of this method. Okay. Now, if your genetic code is made up of 2 bases, that means at any one time, you go into the bag and you try to pick 1 base out. So your base can be A or T or G or C for this one here. So it can be A, T, or G, or C. Then after that, you put the base back there, you mix them, and you randomly pick again. So when you randomly pick again, you can also be picking A or T or G or C. Then you put it here. All right. So then you see what is the base that you have obtained for these two. So there are four possible ways of getting your base here, and there are four possible bases that you can get for this one. So therefore, how many different type of pattern that you can get is actually 4 different type of bases in your bag, and you want to take 2 out to arrange. So it's 4 power 2. So 4 power 2 is 16. Okay. Now, if you say that, okay, now I actually want to take 3 out. So if you want to take 3 out, what are the possible different combination of bases you get at the end of the day? Is to start off with 4 bases in your bag, and you actually want to take 3 out to arrange. Okay, so what are all the possible arrangement of the bases you can get is 4 to the power of 3, because you've got 3 seats here. So therefore it's 64. So this is a formula that you need to memorize. You need to memorize in order to know what is the number. Not memorize the number, memorize the formula. Okay, so this is the easiest way that I find that I can remember this formula. How many different codons, how many different bases I have in the bag, how many bases I want to use to make the code. So it's power how many. Then we'll give you all the different possible combination of the bases for these 3 seats that I have. 3 seats, S-E-A-T-S, seats. Okay. Right, so let's move on. So the code is universal. Universal means the same triplet code for the same amino acid in all organisms, you name it, whether it's plant, whether it's bacteria, fungus, animal, okay, prokaryotes, the same code will code for the same amino acid, regardless of the organisms. So this is something that's very wonderful, okay? Except for a few triplet codes in mitochondrial DNA and ancient bacteria may code for different things. So other than that, the rest code for the same. So it's called universal. The code is universal. So that's why we can use genetic engineering. We insert the human insulin gene to the bacteria, and the bacteria will start to use the gene to make insulin. So we are using bacteria to make insulin for us. It's because the genetic code is universal. So that's so wonderful about the nature. Now, the next property is called degenerate. The meaning of degenerate means one amino acid may be coded by more than one codon. So this is the meaning of degenerate. How do you relate to this word? After I'll explain. There is, because we have got 64 codons, right? 64 codons, definitely, but we only got 20 amino acid. So we've got 64 codons that code for, and we only got 20 amino acids. So definitely there'll be a few codons that will code for the same amino acid. So this property is called degenerate. The codons are degenerate. That means a few codons may be coding for the same amino acids because we got more than enough. Okay. So why is this important? So that when point mutation happen, it do not change the amino acid. Now, point mutation means change in one base. Change in one base. Okay, so this is called point mutation. Change in one base. It will not cause a change in amino acid. So that the amino acid sequence in the polypeptide will still remain the same. So I'm going to use the genetic diagram to show you afterwards. So the tertiary structure of protein will not change. That means even though there's a mistake, okay, in the DNA or in RNA synthesis, there is a wrong base is added in, it will not change the amino acid sequence in the polypeptide. So your polypeptide synthesize will still function normally. So this is something very important. So the protein will function normally. So this is what you can see from here. Okay, can you see that UUC code for phenylalanine? All right, and then you got ACU, ACC, ACA, ACG code for threonine. Then CGCU and all this code for alanine. So you can see that for alanine itself, there are 4 different codons that code for the same amino acid alanine here. Okay, so therefore this is called degenerate property of the codon. Now, why is it called degenerate? Can you see here? For all these 4 different triplet codons, if there's a mistake in the last base, so instead of a U, a C is added in, it's still code for alanine. Or instead of a C, right? If A is added in, it's still code for alanine. Or instead of a mistake occur, G is added in, it's still code for alanine. Can you see that? So this is called degenerate. Degenerate means when you say the last base has changed, right? Has changed to something else, but it's still coding for the same amino acid. So this is a very important property. Can you see for all of them, when you change the last base, it still code for the same amino acid here, right? When you change the last base, it code for the same amino acid. So for all of them, see, you change the last base, it will still code for the same amino acid. So this property is called degenerate. There's more than one triplet codon that will be coding for the same amino acid. So this is called degenerate property. Okay. Now, 4th property of the codon or genetic code is punctuated. That means when you write a sentence, right? When you write a sentence, there's a comma, okay? There's a full stop, right? So this is called punctuated. Now, when we read the genetic code, it must also be punctuated because it needs to tell us, so this is the beginning of the sentence, this is the end of the sentence. So in the genetic code, it must tell us, because there's so many bases in the DNA, right? Which base is the beginning of a gene? Which base is the end of a gene? So it must tell us, isn't it? So you look at this genetic dictionary. This genetic dictionary. Can you see that there is this methionine here that's highlighted? Now, AUG is always the start codon. Start codon is always AUG. So this one, please memorize. And AUG always code for methionine. So the first amino acid is always methionine because the start codon is AUG. AUG stands for codes for methionine. So there's one start codon and there are 3 stop codon here. So UAA, UAG, and UGA are stop codon. So if you want, you can memorize these 3. Very easy to memorize. UAA, UAG, UGA. So these 3 are stop codon. So you can see that out of 64 codons, okay, there is 1 start codon and 3 stop codons. Okay, so therefore, out of 64 codons, how many codons code for amino acid? Let me see first who I want to call. Tan Jiaying. Out of 64 codons, how many codons code for amino acid? Okay, Tan Jiaying is not responding. Can you all please type your answer in the chat? Oh, you're very fast, 61. Okay, correct. Rebecca, why 60 lah? 64 minus 3 lah. AUG code for amino acid, Okay, so Andrea, yes. Andrea is the first one to type the correct answer. So Andrea, you get yourself one point. Okay. Okay, right. 61, right? Because stop codon does not code for any amino acid, right? That's why you stop, isn't it? but start codon code for methionine. So altogether 64 minus 3 is 61. So therefore, 64 minus 3 is 61. All right, so 61 codons code for the 20 amino acids that we have. Now, the codons are non-overlapping. So what do you mean by non-overlapping? So let's say if these are the base sequences in the mRNA, and I want to translate it to amino acid sequence. So how do I read the bases? Always non-overlapping. So once I read AUG, right, once I read AUG, the next base I'm supposed to read is CAA. Then after that is GUA. So this is called non-overlapping of reading the bases. Okay. So this AUG code for the first amino acid, CAA second amino acid, GUA third amino acid. So when you say overlapping, what do you mean by overlap? You read AUG. Okay. After that, you read G again. So G overlap with G already, right? So GCA. Then after that, you read the A again. So the A overlap with the A. So this is called overlapping by one base. And this example is overlapping by two bases. That means AUG, and then you read the UG again here. Okay, UGC. Then you read the GC again here. So overlap. The GC and GC overlap. So the actual way of reading is non-overlapping, which is actually this one here. Non-overlapping. Okay, so this is the property. So altogether 5 properties of the genetic code. Let us recap. Number 1, 3 bases code for 1 amino acid, and the code is universal in all the organisms. And the code degenerate. That means more than 1 codon can code for 1 amino acid. When the last base change, it may still be coding for the same amino acid. Punctuated means there's 1 start and 3 stop codon. Non-overlapping is how we read the bases. Okay, so now we want to look at how transcription can happen. How transcription can happen. Okay, so transcription is a synthesis of mRNA from the gene. So it is a mechanism, means process, by which the base sequence of a gene is converted to the complementary base sequence of mRNA. So this is the definition of transcription. Okay, transcription, you can say it is a synthesis of mRNA. The detail is when the base sequence of a gene is converted to the base sequence of the mRNA. So this is the definition of transcription. Okay. So the steps involved is number 1, of course, this is a gene here. So starting from the beginning of the gene, the DNA must unwind and unzip. So unzip means like this is a zip, you unzip it means you break the hydrogen bonds between the complementary bases so that these 2 strands will separate. Okay. So after unzipping, all the bases are exposed already. So RNA polymerase, so now you want to make RNA, so you use the enzyme called RNA polymerase to make the RNA, okay? So RNA polymerase will use the DNA template strand or the transcribed strand. So out of 2 strands, only one of them will act as a template, right? Because mRNA is single-stranded, so we only need to use one of the DNA strand to act as a template. So over here, you can see that the DNA strand at the bottom, right? The DNA strand at the bottom is used as the template. So this is called the template strand or the transcribed strand. Transcribed means the copied strand, right? So the DNA template strand will be used. So the RNA polymerase will add complementary free RNA nucleotides. So the nucleotide now is called the RNA nucleotides. So this is the enzyme RNA polymerase. This arrow is wrong. Okay, this arrow is wrong because this arrow is wrong because the strand is at the bottom here. So I've changed it. I've changed it already. So the strand is the bottom strand, is the template strand. Okay. Okay, so the new nucleotides are added into the template by complementary base pairing in a 5' to 3' direction. So you see this RNA also grows from 5' to the 3' direction, right? So DNA and RNA both will grow from 5' to 3', right? So once the entire gene has been transcribed, the RNA will then leave the DNA at the end of the gene. So this is the process of transcription, okay? So here, what you can see here, this is a bigger diagram for you to see the complementary base pairing here, right? So if this is a DNA template strand, then this U will pair with A. So U pair with A, U pair with A, G, C, G, C, C, G. And then here, A will come in, right, to pair with T. A and then C. So this is how the nucleotides are being added in by complementary base pairing. Okay. So this is called the non-transcribed strand. Okay, non-transcribed strand. So there is a video that's very interesting that I would like you to watch, right? So this is the YouTube link. You can actually click and watch this, video. Now you only need to watch until transcription. You don't have to watch the protein synthesis because you've not learned protein synthesis yet. Okay, so you can watch it at your own free time. Because now I only left with 2 minutes. I'm not sure whether it's enough for you to watch. So therefore you can watch it at your own time. All right, so that's all for today.